So the first blotting technique that was developed was by Edward M. Southern in 1975. So it is not a very new technique. It is very old technique that was used, but definitely during that time when this technique was evolved or was evolving, today you have seen in our labs also that we have a blotter, electrical blotter, okay, that fastens the reaction, which uses the electric current. But earlier, this entire method was dependent upon the sole blotting method, or you can say the siphoning method of your tissue papers. Okay, so blotting technique is an hybridization technique for identification of a particular size of DNA. So now blotting is being applied in various different types of macromolecules. You can do it for DNA, RNA, as well as proteins, right? So, but this particular method was developed for DNA, and so the from the name of the developer or your scientist, that is Edward M. Southern, the Southern blotting technique, it was named. Okay, but now we also have Northern technique, we have Western technique, which is for your RNA and proteins respectively. Okay, so this technique is based on the principle of separation of DNA fragments by your gel electrophoresis first, and then once it has been separated by electrophoresis, then these fragments are going to be transferred onto a nitrocellulose membrane, and then identification of these fragments. Okay, so which is a gene of interest, we have to understand, right? So the identification would be using your labeled probes. Okay, so that labeling can be done either by radioactivity. Earlier it was through radioactivity only, but nowadays we use fluorescence technique, as radioactivity is not very safe for the users. Yes. So basically the DNA fragments are separated on basis of size and charge during electrophoresis, and separated DNA fragments after transferring on your nylon membrane, that is your whatever, any membrane here which will hold on to these macromolecules that is being used. Yes, you have already seen a nitrocellulose membrane. It looks like a filter paper. Yes. So the desired DNA is detected using specific DNA probes that is complementary to the desired DNA. So hybridization probe, you all know that it is nothing but it is going to be a short artificially synthesized single-stranded DNA, which is going to be complementary to your gene of interest, right? So the probes are usually been labeled by a marker, as I said, either it would be a radioactive marker or a fluorescent marker. So let us now understand the steps or the procedure of this particular method. So first is restriction digestion. So because when you are isolating, we have seen that what you are going to isolate the whole genome of that particular organism, right? So once you have isolated the whole genome, now it is your responsibility that you need to just identify or just isolate your gene of interest. So next step, what you are going to do is your restriction digestion. Okay. So mostly what you will use is your restriction enzymes. See, whenever you are working with any kind of your sequences, you have to first understand the sequence, and whenever you go for your gene of interest or anything, we would be knowing the sequence of the gene of interest, and also what are these sequences which are flanking these sequences, that also would be known. So what happens is you are going to use that kind of your restriction enzymes which are near about your gene of interest, right? So, but these restriction enzymes are not going to be present only at those regions, it will be present somewhere else also. So what will happen is it will be giving you many fragments, and from that fragment you have to identify your gene of interest. So you do your amplification of that particular fragment, you will do your restriction digestion, and then you will do your gel electrophoresis so that you get your proper band. Once you get your proper band, so now this is your double-stranded DNA. So it has to be denatured. So either treating with your HCl and NOH will help you in denaturation of this particular DNA fragment, and then you blot it. That means you transfer this DNA from your gel onto this nitrocellulose membrane. So once it has been transferred onto your nitrocellulose membrane, it has to be baked and blocked using casein in BSA. So baked and blocked in the sense, when it is being washed, the nitrocellulose membrane is being washed, this DNA, which single-stranded DNA, which is now been transferred onto your nitrocellulose membrane, should not get washed away. It should become stable. It should become etched upon that nitrocellulose membrane itself, right? So that is an important part. So then you add your hybridization probe, then you give a washing, and then find out whether the radio or autoradiogram is being developing or not. So if you get proper spots, that means the gene of interest is present onto that particular blot. Okay, so that will help you in which position in the DNA. So if you are running the gel, at what position you are getting that particular band, that will help you in identifying the fragment, which is the fragment which is necessary. Okay, so this is your southern blotting method. Sorry. Method that I was discussing about. So you can see here, this is your gel electrophoresis, whatever your labeled RNA or DNA of known sizes serving your size markers, and then this is the arrangement that is going to be done. Okay, so that you will have, you have seen the blotters, right? You have this sponge and you have these filter papers, and uski beech mein you have this the gel, and on top of the gel, just on top of the gel, you would be having your nitrocellulose membrane. So the sponge and this paper towels or your tissue papers, they help in absorbing the liquid, so that by siphoning action, and always the top layer of your paper towel should be dry. So that it is because of the siphoning action, what happens is the water molecules which are present in the buffer. Here you can see that this is the tray which is containing the buffer. So what happens is when this water molecules are traveling from this from the sponge to your this paper towels, what will happen is in between you have this sandwiched gel and your nitrocellulose membrane, right? So water molecules will press from the sponge. Now it reaches to your agarose gel, and agarose gel is a porous thing, right? So it will force, so from this force, it will force this DNA molecules to go upwards, to move upwards. And as it moves upwards, what is present is your nitrocellulose membrane. Now the nitrocellulose membranes are going to have the pores which are very, very small. Okay. So even if now the water molecules are pushing it up, more up, this DNA cannot be coming out of this nitrocellulose membrane, whereas the water molecules can come. So just imagine नीचे ज़्यादा पोर वाला है और ऊपर less पोर वाला है. So when the water molecule is pushing through this pores, what will happen? The DNA would be pushed out of this pore from where the pores are bigger. Okay, it will be pushed out, and then it will stuck into this. It will get stuck into this nitrocellulose paper, and further, still pushing is going on. But nitrocellulose paper is having small pores. Hence, it will not be able to move away from this nitrocellulose membrane, and it will get stuck onto this. Okay. So here you can see once it has been done, so for a period of time, so your nitrocellulose membrane is then removed, which is now is going to have your nucleic acid bound to it. Once that has been done, then you keep it, so you can keep it in your buffer so that it doesn't dry up and it is present, and then the baking is being done, and treatment with DSA and casein is being done, so that your non-specific sites are not present where your radio labeling will go and attack. So then you will allow the radio label probe to hybridize to the separated DNA, and then you are going to get this. So whatever your bands, if you get, so labeled bands you get, that means you can consider the positioning from your, so this is your marker, right? Your labeled marker. So that will also help us in understanding what is the molecular weight of your fragments that you are going to get, and from that you will be able to understand. So later on, when you are using the same sample with the same restriction enzymes, the same positioning or the same fragments would be your gene of interest. Okay, so this is the principle. This is the principle of your of your this entire blotting method. So the same thing, let us quickly go through the stepwise manner. As I told you that first is your restriction digestion. So the DNA has to be fragmented by using your suitable restriction enzyme. And this restriction enzyme will cut at specific sites, generating many fragments. And this fragments, the number of fragments would be then amplified by PCR. Then you will go for gel electrophoresis. We will be also discussing about the principle of gel electrophoresis and everything in the next lecture, because that's also there in your syllabus, but you already know it. So whatever your fragments that you have obtained from this restriction digestion, that can be separated by gel electrophoresis. Then you go for denaturation. So SDS gel can after electrophoresis is then soaked in alkali or your acid, that is your HCl, to denature the double-stranded DNA fragments. So now you can see here something written as your SDS gel. So you would be wondering why we are not using agarose and why we use SDS gel. It is there is some notion among the students that SDS stage can be used only for the proteins and not from the nucleic acids. No, it is not that way. See, what is more efficient, or talking about the pore sizes. So SDS gel can give you a range of pore sizes which we may not be able to have in agarose. Okay. So even if we are changing the percentage of agarose, we will not be able to control the pore size of agarose as efficiently as we are talking about your SDS page. So SDS page will give you, or polyacrylamide page will gel will give you more specific pore sizes, and that will help us in separating the fragments more efficiently. Okay, so that can also be used for your nucleic acids. So the gel is then soaked in alkali and acid, so that the double-stranded DNAs are then getting converted or getting denatured into your single-stranded DNAs. Okay, and then the next steps will be moving on. So that is your blotting. So once the separated DNA is there, as I said, it will transfer to positively charged membrane, nylon membrane, that is your nitrocellulose paper, by the process of blotting. So process of blotting, I just have explained to you how the water molecules are moving. And this movement and all will be definitely being facilitated by this electric charge that is being produced. So then you have the baking and blocking. After the DNA of interest bound on the membrane, it is baked on autoclave to fix it in the membrane. The membrane is then treated with casein or BSA, which saturates all the binding sites of the membrane. That is what I was talking about. So that these sites, which are other sites other than your nucleic acids, whatever your remaining sites are there, if they are kept open, so sometimes this probes can go and bind to this other sites. Okay, because nitrocellulose membrane has an affinity for this nucleic acids, and the probes are also your nucleic acids. So even if there is no DNA, these probes can go and non-specifically go and bind to these places on your membrane. So that is the reason why this BSA is being utilized, or casein with BSA is being utilized, so that these binding sites are blocked. So when you are adding the probe, they cannot go and bind to these sites which are present. So the DNA bound to membrane is then treated with label probes. The label probes contain the complementary sequences to your gene of interest, and the probe bind with complementary DNA on the membrane, since all other non-specific binding sites have been already blocked by BSA or casein, right? So then last step is the membrane-bound DNA labeled with probes can be visualized under autoradiogram, which will give you the pattern of the bands. And from the pattern of the bands, you can identify what would be the size of your gene of interest. And accordingly, you can then further separate out that particular band after doing the gel electrophoresis of your restriction digestion. So this is the same thing as I said. First, genomic DNA restricted, getting fragments, agarose gel electrophoresis. Depending upon the size of the fragments, they will get separated. Then you will go for blotting, so that this brands from your agarose gel that transfers into your this nitrocellulose membrane, and then you can just treat it with the labeled probes, and you will be getting darker bands when exposed to X-ray film. That is your autoradiogram. Okay. So, is this technique very much clear to you? You have already done Western blotting, so you know this technique, right? So, is this technique clear to you? A quick yes or no? Okay. Okay, great. So, let me just move forward. So, talking about the applications of southern blotting, the southern blotting technique is used to detect the DNA in given sample. Yes, then DNA fingerprinting is an example of your southern blotting. Yes, so DNA fingerprinting, what we do where we understand that there are different like you go for some marker DNAs, right? So when you are separating, you get a pattern, and that is this entire process of DNA fingerprinting is going to be dependent upon your southern blotting technique. So it is used for, so when you have DNA fingerprinting, definitely all the applications of your DNA fingerprinting can also be considered as an application for your southern blotting. So it can be used for paternity testing, criminal identification, victim identification. Yes, all which are your applications of your DNA fingerprinting. Then for your cloning purpose, it is to isolate and identify the desired gene of interest. Okay, used in restriction fragment length polymorphism. Yes, RFLP. For that also, just as we said about DNA fingerprinting, so in that also RFLP is one of the markers. So similarly, not only for humans but for other different types of genes also, we for understanding them, for studying them, RFLP can be used to identify mutation or gene arrangement in the sequence of DNA. So the probes that you are going to use should be specific for these mutations or gene arrangement. So if the probes go and bind, then you can identify that yes, this particular mutation is present in your genome. So used in diagnosis of diseases caused by genetic defects, as I told you, again you have to use specific probes for that particular genetic defects, and you would be able to identify it. And lastly, you can also use this for identification of your infectious agents. Okay, clear to you? Southern blotting, any doubt?